हेलो फ्रेंड्स, मैं ध्रुव कुमार। आज इस हम सुपरफास्ट रिवीजन सीरीज में एनसीईआरटी बायोलॉजी कवर कर रहे हैं। इसमें आज के टॉपिक है मॉलिक्यूलर बेसिस ऑफ इनहेरिटेंस। सो स्टार्ट करते हैं हम लोग। मॉलिक्यूलर बेसिस ऑफ इनहेरिटेंस, ठीक है। तो आप देख सकते हैं इसे। जेनेटिक्स, व्हाट इज जेनेटिक्स? Genetics mainly deals with the study of genes, heredity, and genetic variation. Genes exist on chromosomes, and chromosomes are comprised of DNA and proteins. DNA is a molecule that carries genetic information in all living organisms and viruses, where it is used in reproduction, functioning, growth, and development. It is a long polymer of न्यूक्लियोटाइड्स। ये आप देख सकते हैं ह्यूमन हो गया। इसके अंदर जो सेल्स होते हैं, सेल्स, इसके अंदर क्या है, ये क्रोमोजोम है, और क्रोमोजोम के अंदर क्या है, ये डीएनए है, और डीएनए के अंदर ये है हमारा एक न्यूक्लियोटाइड, और ये पूरा क्या है, जीन सेगमेंट, जीन है, डीएनए ऑफ सेगमेंट, ठीक है। और ये डीएनए इज a double helical structure. DNA is a double helical structure that carries all the genetic information. Its length is determined by the number of nucleotide pairs present in it. It is an acidic substance in the nucleus identified by a Friedrich measure. It is a double helical structure given by the Watson and Crick, ठीक है। तो ये हमारा जो है DNA का डबल हेलिक्स मॉडल है। इसमें क्या है, एडेनिन, थाइमिन, ग्वानिन एंड साइटोसिन्स है, ठीक है। सेंट्रल डोगमा, the central dogma was proposed by Crick. The central dogma states that DNA is converted into RNA and the RNA is converted into protein. So DNA is converted into RNA, and RNA converted into proteins, ठीक है। So it is a central dogma. In retrovirus, the flow of information is opposite: RNA to DNA, DNA to messenger RNA to protein, ओके। ये DNA का स्ट्रक्चर हो गया, RNA का, और ये प्रोटीन। DNA to RNA ट्रांसक्रिप्शन होता है, और RNA to DNA, RNA to प्रोटीन, DNA to RNA ट्रांसक्रिप्शन, RNA to protein है ट्रांसलेशन। DNA packaging. The negatively charged DNA is packed by surrounding the positively charged histone octamer. A structure called nucleosome is formed. The DNA is packed in a chromatin of eukaryotes. That is naked. That's the DNA. Active genes, nucleosomes, chromatosomes, less active genes, solenoid, and 30 nanometer fiber. Interphase में chromatin and metaphase, that is a chromosome. DNA replication. DNA is a self-replicative. It occurs in S phase of the life cycle. It takes only a few minutes in a prokaryotes, but hours in a eukaryotes. DNA undergoes semi-conservative replication. That's two strands of DNA are formed. One strand is same as the other strand, while the other is complementary to the parent strand. The replication occurs in 3 to 5 directions. Summary of DNA replication है, आप देख सकते हो उसे। Parental DNA, yes, complete structure. RNA. What is RNA? RNA is a ribonucleic acid. RNA is a vital molecule that are with a long chain of nucleotides. It is the first genetic material. Our nucleotide chain comprised of a phosphate, ribose sugar, and nitrogenous base. RNA acts as a catalyst and as a genetic material. There are two types of RNA, that is a genetic and non-genetic. This is a DNA, this is a RNA. That's the structure. Okay. Human Genome Project. The Human Genome Project launched to sequence the entire human genomes of 2.75 billion base pair. The main goal of the Human Genome Project are to provide a complete sequence of 3 billion base pairs that make up the human genome, to sequence the genome of other organisms that are used in medical research for mouse and flies, to develop new tools to obtain, analyze the data, and make this data widely available. It holds prospects of a healthier living, a database of knowledge about designer drugs, genetically modified diets, and genetic identity. This is a human genome project. Human genome project. इंटरनेशनल इन्वॉल्वमेंट है इसका, नेकेड जीन्स ये हमारे जीन्स का, ठीक है, डायग्नोस्टिक टेस्टिंग, राइट ड्रग्स फॉर राइट प्लेयर एंड जीन थेरेपी, ये सब में इसका देखिए. The DNA sites where DNA dependent RNA polymerase bind for the transcription is called answer operator, promoter, regulator or receptor. So answer is B. That's a promoter. DNA binding sites. DNA binding sites क्या? DNA binding sites are a type of binding site found in a DNA where other molecules may bind. DNA binding sites are distinct from other binding sites. They are part of DNA sequence. They are part of DNA sequence, that is a genome, and they are bound by DNA binding proteins. DNA binding sites are often associated with specialized proteins known as transcription factor, and thus linked to the transcriptional regulation. The sum of DNA binding sites of a specific transcription factor is referred to its system. DNA binding sites also encompasses the targets of other proteins like restriction enzyme, site-specific recombinase, C site-specific and methyltransferase. This is a promoter recognition sites. ठीक है, transcription transcript region for DNA. DNA polymerase single stranded template DNA. Operand model for the regulation of transcription was proposed by whom? Michelson and Stoll, Jacob Monod, Watson Crick, and Horsey and Chase. So answer is B. That is a Jacob and Monod. The operand model of a prokaryotic genes regulation was proposed by Jacob and Monod. Groups of genes coding for the related proteins are arranged in a unit known as operands. The operands consisting of operator, promoter, regulator, and structural genes. That's a diagram. The central dogma of molecular biology, genetic information flow, was modified by whom? Modified by the discovery of what? DNA polymerase, DNA ligase, reverse transcriptase, and DNA polymerase. Answer is C. Reverse transcriptase. The human chromosome with the highest and least number of genes in them are respectively chromosome 21 and Y, chromosome 1 and X, chromosome 1 and Y, and chromosome X and Y. Answer is C. Chromosome 1 and Y. Human genomes. Human chromosome. Chromosome is an organized package of DNA found in the nucleus of the cell. Different organisms have different numbers of chromosomes. Humans have 23 pairs of chromosomes, 2 pairs numbered chromosomes called autosomes, and 1 pair of sex chromosomes X and Y. Each parent contributes 1 chromosome to each pair so that offspring get half of their chromosomes from their mother and the half of their father. The structure of chromosomes. Okay. सेल हो गया, न्यूक्लियस हो गया, इसमें किसी में क्रोमोसोम. क्रोमोसोम का एक क्या टेलोमेरेज हो गया, ये सेंट्रोमियर हो गया, ठीक है, ये सिस्टर क्रोमेटिड हो गया, ये शॉर्ट आर्म हो गया, ये लॉन्ग आर्म. Which of the following are the functions of RNA? RNA का क्या फंक्शन? First, it carrier of the genetic information from DNA to ribosome synthesis, polypeptides. It carries amino acids to ribosome. It constitutes components of ribosome or of the above. Answer is D. Primary functions of RNA. RNA का primary function. The primary function of RNA is to create protein via translation. RNA carries genetic information that is translated by ribosome into various proteins necessary for cellular process. Messenger RNA, ribosomal RNA, and another 3 main types of RNA involved in protein synthesis. RNA also serves as the primary genetic material for a virus. Other functions including RNA editing, gene regulation, and RNA interference. These processes are carried out by a group of small regulatory RNA, which include a small nuclear RNA microRNA and a small interfering RNA. This is a messenger encoding proteins. This is a tRNA act as a adapter between a messenger RNA and amino acid. This is a ribosomal RNA forms the ribosomes. Okay. So friends, आपसे रिक्वेस्ट है कि प्लीज इस चैनल को शेयर सब्सक्राइब कर दीजिए, लाइक कर दीजिए और शेयर कर दीजिए. थैंक यू.